is Krzysztof Wabnik from, he's a group leader at the CBGP at UPM, that's the Polytechnic University in Madrid. And he's going to talk about robust frequency-encoded dynamics in a minimal synthetic phytohormone system. So Krzysztof, the stage is yours.
All right, thank you, Phil, for the introduction and invitation to this mini symposium. Hopefully you can see my PowerPoints.
Yes.
Does it work? Okay, perfect. Okay, so as already Philip mentioned, I'm gonna show you a part, well, small part of the research we do in our lab related to phytohormones. And phytohormones are essentially the plant hormones, and their role is to regulate the development and growth of the multicellular organs, pretty much like in animals. Okay, first of all, well, I have to acknowledge people who work on that project, and this data I'm presenting is unpublished. So there are in particular two of my students. It's a bachelor student, Sarah, who started the project, and with the help of Mario is now joined the PhD program. So he is a PhD student, but before he was master. So these two guys essentially contribute a lot, and I have really, I'm really happy to have such a great team of scientists coming from different fields. So our approach to introduce you into the problem is to combine computer modeling, and the computer modeling also the level of single cell or the level of the whole organ, combined with microfluidics that Henrike mentioned in her research. And we study dynamics in the simpler organisms where we can actually decouple some of the processes. It's example of growth of one of plant organs called hypocotyl. We can dissect some mechanisms that govern this growth using some simpler organisms like yeast. Okay, so why we are interested in hormones, and in particular plant hormones, so phytohormones. And the reason is because there's one of them called auxin, from Greek it means to grow. And basically this hormone regulates almost everything you can find in plants, starting from regeneration of tissue after wounding, early in the embryogenesis, and post-embryonic development, which is formation of new organs, something that plants are really unique about. So I'm coming from plant field, so my interest is additionally, as you can imagine, it's close to my heart. So we study in particular plant hormones. But it's a more general problem. I think I just switched from the full screen, sorry. So the more general problem is that the hormone sensing in multicellular organisms involves usually a cascade of events. So for example, on this picture that I'm showing you here, you can see that auxin, it's a very small signaling molecule, small chemical. It requires co-receptor system, which includes a number of components of ubiquitination system in plants. And then it recruits specific repressor of signaling, and this repressor is degraded. So then you can get a response from transcription factors called auxin responsive factors. So this cascade is quite complicated, and it's not easy to work on it because there are many of those genes duplicated in plants. So it makes it very complicated. So one of the ideas we have to try to port this system into simpler organisms like yeast, which is still eukaryote, so we can expect similar transcriptional machinery operating there. But on the other hand, we are thinking to try to look for alternatives, alternative receptors for that particular hormones that could be used without the need for additional components. Similar situation happens in animals where you have, for example, steroid hormones. They also have to travel a long way before they can actually affect the transcription of downstream genes. And this is typically the translocation from nucleus to the cytoplasm, and then you get specific expression or downregulation of the gene, depends on the context. So we are interested in the simpler system, whether we can build such. So we look on the microorganisms that are associated with plants, and there are many of them. It's a whole microbiome that basically sits on plant, and it can be beneficial or pathogenic to plants. And many of those organisms turned to sense hormones. And the question was how they do it, right? And there's a specific family of transcription factors from that bacteria. It's called multi-antibiotic resistant genes. They're quite known family of stress sensors. So they stress antibiotics, well, they sense antibiotics, and they provide a stress response to bacteria so they can adapt their response and growth. Some of them were crystallized, so you can see the crystal structure of those, but really the actual mechanism is not really well understood for those. Question was whether we can find those MarRs that can sense plant hormones such as auxin, and whether when we transplant such a system in eukaryote, it would actually work, right? So first we identify two potential MarR genes from different bacterial species. One of them is E. coli 1 that is quite known, and it senses salicylic acid in the way that salicylic acid inhibits its function. I will tell more about the mechanism later, but what is important, we actually, typically these guys are repressors in bacterial system. But now when we introduce the system into eukaryotes, in this case yeast, we drive it from sugar-induced promoter, and we turn it into activator by tagging a small VP64 activation domain. And then we know more or less what the operator sites are for this guy. Then we can put fast recycled reporter, which is in this case, we call it the GFP, it's essentially ubiquitin tag GFP, which allows for recycling. So there's high degradation rate, so we can increase the dynamic range of this system. Similar we did for the another protein that was actually not known to sense IAA. There was a proposition it may from one of the plant pathogens. And actually we were really excited to test whether this would do, and this would do in the eukaryotic system. Similar scheme, again, galactose as the inducer, VP64 tag to our protein. They are quite small proteins, so easy to work with. And then we don't know exact operator site, but we know the genomic region that has the operator site. So we use that. It's not big, it's around 200 base pairs. And first of all, we check whether we have a sensing, and for the auxin sensor, we get quite good kinetic curves. We can see 2 order of magnitude recognition of concentration, which is in physiological range in plants. So in fact, this sensor can be used as a sensor in plants. And it's a direct sensor because auxin binds directly that protein, not like in the system that I showed you in a minute, where you need a whole cascade of events to for auxin to be able to regulate the downstream process. For the salicylic acid, which is another plant hormone important for immunity, we see also twofold dynamics, but somehow it's less sensitive to the IAC-R, which is the auxin sensor. However, it's still within the physiological range, so we can use it both in yeast and in plants. So the first idea was to essentially, because these are static measurements, of course, so they don't give us information about dynamics of this system. So as you may imagine, Spain is not particularly rich in terms of financing science, so we have to find some other ways to be able to do fancy experiments. So we developed a method where we can actually design the microfluidics devices using thermo compressed sheets of plastic, where we can actually design the the device. So you just shrink this. From that, you can get the, well, this is your mold. From that, you get microfluidic device. And then we have a customized gravity-based hydrostatic pressure flow system that we develop in the lab to be able to control the inputs into the microfluidic streams. So the advantage of this method is you don't need any clean room or specific equipment to do this. You can do it on your lab bench, and you can do it in a matter of hours. Go from design, actual device running experiment. Of course, this is not as precise as other systems, but for our purpose is essentially enough. We have mixers on our chips, for example. They have a very good mixing ratios as expected. We have additional chaotic mixers to ensure that the fluids are well mixed, and then trapping regions where we actually can, supply cells, and they can happily grow in those regions. So we use that system and we run long-term experiments, and we see what is the actual response from those two receptors. And we see that, so if this is the stimuli of the hormone that we provide to the microfluidic system, we got this kind of response, which was reproducible in many, many traps and experiments that we did. Interesting aspect here was that the response was delayed. So it was not exactly tracing the input that you put in the system. So that was already interesting. And actually, you can characterize this kind of behavior by looking on input versus output relation. And you see basically the difference or the delay in the response as a measure of precision in this system. So we could measure the precision of these two sensor systems. And if we have, obviously, if we have delay, we have some kind of memory effects integrated in that system. And the precision was not perfect, so that suggests that there is some delay in the response in such a simple system, as you can see. Then we look into the structural information, because there's actually resolved crystal structure for MarR, the one that sends acetic acid. And we asked from where we could get this delay. And one of the hypotheses we had, maybe it's encoded in the structural information, so structural changes. So the apoprotein means it's without its ligand, so without acetic acid, it's pretty much unstable. So it kind of closes, open, and it's like continuing that cycle. But then in the DNA bound configuration, where it's your operator, it's very stable. It maintains the distance between two monomers. It acts as a dimer, and it's pretty stable. So it could give information that there is a stable and unstable state in the system. Another thing is that we try, so we just summarize that. So we have this oscillatory conformational dynamics at the protein level, and then we have more like open conformation in terms of DNA bound protein. But then with the whole protein, something interesting happens. Actually, the whole dimer closes. So we have two molecules of acetic acid here. So this molecular dynamic simulation, you can see that they close and then they maintain that state. So it could be another stable state in the system, right? Then, so it's preferentially tends to the closed configuration. And then, well, we can analyze the system by looking on the evolution of dynamics of the distance change between the monomers. And you can see the apo is pretty unstable. Then the holo or DNA bound, they tends to be more stable in terms of conformational dynamics. And again, we can plot the probability or frequency of observing in our simulation, observing distance between DNA bound and holo. You can see that there are just intermediate of the Apo configuration, which can go from one state to the other. So we think we have typical bistable system where DNA bound configuration gives us the stable state where we have the actual response. And then Apo just travels between two depending on the actual hormone concentration, which can put it in the holo configuration. Okay. We run also metadynamics. I'm not gonna go into the details because we don't have really time for that. But the idea here is whether we can perturb the system from DNA-bound configuration in particular, whether it will come back to that state or it will explore other possibilities. And we found that in terms of DNA-bound, of course, it will stay in the same state because we perturb, but it still goes back to the same state. The same happens for APO. So APO can actually reach the DNA-bound state as one expected, but it doesn't happen in holo configuration, where we tend to have shorter distances. It's enabled to bind DNA. So what we think could explain this delay or memory effects, transient memory, is that we have two states which are stable, which is hormone bound and DNA bound, and then unstable state, which can go in one of those states, and you can actually push the system toward one of the states using the hormone. For the IAC-R, so the oxygen sensor, we don't have structural information. We run some models and we see similar kind of tendency, at least at the qualitative level. But again, it's just the simulation. So we don't have actual crystal structure to be able to do that. Then we thought, okay, what happens if we combine both? Okay, so we left IAC-R regulated by auxin as an activator, and then we put the other one as a repressor in the system. And you can do it just by, instead of VP64, we plug repression domain from one of the yeast proteins, yeast repressors. And interestingly, well, we could see the logic operation that this circuit can perform depending on the state of the hormones, how much of the hormone you actually apply, whether it's SUST, GEST, or OX. We see similar thing under microfluidics, where we can also observe those 4 states. It's called NIMPLI logic gating, and we are also exploring other type of gates using that system right now, but I'm just, for sake of simplicity, I'm just showing you one. Then when we run dynamics, because that was kind of static measurements where we put the different hormones in the multi-well plate and then we measure the response, what happens in the dynamic cycle? So when we stimulate system with both hormones with 10 hours period, so we have 10 hours clock that stimulate that system, then we got delayed response, and then delayed response was exactly 3 times the input period. So that was interesting. So when you look at that, so for each of the pulses of the hormones, we could see the response exactly after 3 cycles of the inputs. So that means that this circuit, this simple circuit, which has basically repression activation, can operate as a counter. And how that would be. I just show you that any of these systems at the protein level can operate as a bistable switch, okay? So if some of you are familiar with electronics and development of electronic circuits, if you combine 2 flip-flop systems, so 2 bistable systems in a sequence, you can generate counters, okay? And potentially this is what we are observing here. So this is like for different traps that we look, so it's kind of repeatable, and we can see 3 to 4, so it counts to 3, between 3 and 4 essentially for the input. Now, what happens if we speed up our stimulus, okay? So we thought, okay, if we speed up in the simple system, since we don't have feedback loops, at least at the level of the circuit, but we have feedback loops at the level of the, feedback loops. We have basically bistable system at the structural level. So if we speed up the input, what happens? One of the prediction was it will speed up again, but question is whether it still counts to 3, right? So it keeps the same type of memory. So instead of 10 hours, now we stimulate the system every 2 hours, okay? And what we observe is indeed increase of the frequency of the output oscillation. But again, when we look more closely on the dynamics, we did, for example, autocorrelation function, so to show that we have actual oscillatory response in the system, again, like, across the traps, we get around 5-6 hours. So again, this circuit counts to 3, independent of the actual frequency of the system, but operates faster, okay? But there's a couple of things. Well, this again to show you it's 2.5 to 3. That's a typical response versus the input of the signal. But what we see is actually by increasing the frequency of stimulus, this circuit becomes more precise. So that one would expect it could lose this memory effects because we exchange hormones more frequently. And then if you imagine the structural changes also are faster, right? So that explains why the system goes faster and also reduces this memory effect. So the circuit becomes, there's still memory effect, but circuit's much more precise. So we go from 10 hours, 5 hours, and then 2 hours. Okay. Then we look on the amplitude of the system, and amplitude goes down. So if we increase the stimuli, the amplitude goes down by around 25%. The period is of output, so the GFP that we are measuring is actually more reproducible than in case of 10 hours. So the speeding up the stimuli makes system more precise, in fact, in response to the external clock. Precision increased by 15%, period is more consistent. And then when we look on coefficient of variation, we can see that except of amplitude, both the period and precision, so we have less variation here. That means the system is more reproducible in its behavior. So that means that there's more precision in that system. So there's interesting dynamics that I want to show you here is at the structural level. So it's very simple synthetic circuit that I showed you, just with the activation and repression, but it's more complicated than one would thought. Actually, something happens internally at the level of the structural changes of the protein when hormone regulates them. So from this point on, we explore system further. We introduce the feedbacks, negative and positive feedbacks in our circuit. And we were hoping to generate free running oscillations. That didn't happen. But instead we have excitable system, which we think it's perhaps even better for our purpose, where we want to use further this system to control, for example, two populations of yeast strains, one, for example, killing the other, and then we could engineer some interesting dynamics based on excitation through the external signals. So yeah, here I would like to stop and looking for your questions. Thank you.
Thanks, Krzysztof. I'm clapping. Yeah, thank you very much for the nice talk. We're running a bit later, so and nobody asked a question on the Q&A, so maybe let's keep it to one short question. From myself, so if that's the case, and there is really some kind of counting going on, how come all the cells are so perfectly synchronized? Because you would imagine that due to the stochasticity of gene regulation, yes, individual cells might be counting and might somehow be frequency locked, but you would get different locking to different absolute delays.
Yes, I don't know why, but they are really nicely synchronized. Well, not all of them, because not all of them respond, you can see that.
Yeah. Mm.
So the large majority of the population responds it could be due to the design of our microfluidics device, where the mixing is very efficient. So we actually change it, there's not too much delay by changing the hormones, but I don't know. Maybe there's something else in the system that we'll try to explore to explain why it's so well synchronized. And we see it for different, different frequencies of the stimuli.
Yes,
and
yes.
I can tell you one thing. So if we introduce now feedbacks in that circuit, it actually the precision is close to 100%. So I mean, you get impulse and then you get response pretty much without much delay.
Okay.
But I don't know. I mean, it looks like simple system, but it's not.
Yeah.
Already at structural level, it seems that something is happening and we don't really understand that yet. Okay?
Yeah. Yeah, there must be a lot going on under the hood. Certainly an interesting thing to model for dynamical systems people.
I mean, obviously, when you change system towards feedback loops, closed feedback loops, you would expect better precision, but also another thing is that you can speed up the system. It stops counting because you can, you have another level of regulation there that you introduce, right? So you can fine tune better the system. And this is actually what happens. We were stimulating with 20 minutes and it was responding every 20 minutes.
Yeah.
So,
okay.
Yeah, we are playing with the system. There's many unknowns, but it looks interesting. And especially the applications look interesting because we try to control population with that system. And also we are thinking to play with electrical signaling using that type of system because it's quite fast, especially the feedback version, where we could, and we know it's excitable system. So, and that could be some kind of prototype neuron thing that we could try to build.
Great. Thank you very much again.